All right, these are notes that are going to go into your HBS course file. I have them in with the digestive system, although honestly I gave them their own little sticky note. We associate enzymes with the digestive system. Honestly, enzymes are important in a lot of reactions within our bodies, but usually talking about them during digestion makes the most sense. All right, so this is my little foldable. I've got it folded into threes. You'll see here in just a second. So, enzymes are proteins that act as a catalyst to bring about a specific biochemical reaction. Enzymes are proteins that act as a catalyst to bring about a specific biochemical reaction. So enzymes can regulate the rate of chemical reactions in cells. Enzymes regulate the rate of chemical reactions in cells. Catabolism is a type of reaction that breaks things apart. Anabolism brings them or bonds them together. Catabolism breaks apart. Anabolism bond together. So here's my little picture. This piece represents an enzyme. This piece represents a substrate. The substrate bonds to the enzyme, and in a catabolic reaction, the enzyme breaks apart the substrate into products. So in catabolism, we're breaking it apart. Here's the enzyme, here's the substrate bonds with the enzyme, then the enzyme breaks the substrate apart into products. For anabolism, here's my enzyme. I have substrates that bond to the enzyme, then the enzyme bonds the substrates together to form a product. So substrates bond to the enzyme, then the enzyme bonds the two different substrates together to form a product. The substrate is the substance that the enzyme acts upon. The substrate is the substance that the enzyme acts upon. And so I've just got that folded, and I've got it taped right on this corner and taped right on that corner right there so that it's taped in. All right, so another visual. The green part is the enzyme. The red part is the substrate. And I've highlighted this area right here. This is the active site. The active site is the region of the enzyme where the substrate or substrates bond and the reaction takes place. The active site is the region of the enzyme where the substrate or substrates bond and the reaction takes place. Enzymes and their substrate are considered lock and key. So there are specific substrates for specific enzymes. So like a lock and a key, there's a specific key that's going to fit into a specific lock. Induced fit is a situation where the substrate is interacting with the enzyme, and the enzyme changes its shape just a little bit in order to create a better fit to that substrate. So induced fit, the substrate enters the active site of the enzyme. The enzyme clamps down around the substrate, forming an induced fit. So basically, when the substrate bonds to the active site, it's just changing the shape of that enzyme just a little bit to make a more perfect fit. Inhibitors. An inhibitor is a substance that slows down or prevents a chemical reaction of an enzyme. An inhibitor is a substance that slows down or prevents the chemical reaction of an enzyme. We actually have already talked about an inhibitor. We talked about that with blood pressure. So we looked at ACE. ACE, angiotensin converting enzyme. ACE creates angiotensin 2 from angiotensin 1. Angiotensin 2 has other effects on the body. For example, it constricts blood vessels, which helps to increase our blood pressure. So ACE is part of that negative feedback loop that we looked at in PBS. It's angiotensin converting enzyme. It creates angiotensin 2 from angiotensin 1. So ACE helps to raise our blood pressure. When your blood pressure gets too low, it helps to raise the blood pressure. It constricts blood vessels, which raises the blood pressure. So here's an example of an inhibitor. So an ACE inhibitor is a competitive inhibitor which does not allow ACE to change the substrate angiotensin 1 to angiotensin 2. So ACE inhibitor, it inhibits, it stops something from happening. It's a competitive inhibitor which does not allow ACE to change the substrate angiotensin 1 to angiotensin 2. So competitive inhibitor. A competitive inhibitor is one that's going to be right in the way of the active site. So this little blue teardrop looking thing, this is an example of a competitive inhibitor. It is interfering in that active site so the substrate cannot bind to it. Competitive inhibitor interferes with the active site of the enzyme so the substrate cannot bind. The competitive inhibitor is competing for that particular spot. A non-competitive inhibitor, notice that the yellow one's non-competitive, it's not getting in the active site, but when it bonds to that little notch at the bottom, that relationship, that bonding, changes the shape of the enzyme. Changing the shape of a protein changes its function. So by changing the shape of that enzyme, now the active site is no longer available to accept the substrate. So it is non-competitive inhibitor because it's not competing for the active site, but it is still an inhibitor. It's still inhibiting that enzyme from working. The substrate cannot bind because the non-competitive inhibitor has changed the shape of that enzyme. Coenzymes. Coenzymes. The blue piece is a coenzyme. It is allowing this enzyme to accept this substrate because it's changing that shape just a little bit. These guys have to work together in order to bond to the enzyme. Coenzyme is not a substrate. This is still just a substrate, but it requires that coenzyme in order to bond with the enzyme. So the blue piece represents the coenzyme, and it's required for that enzyme to function. For a coenzyme, this blue piece is representing a coenzyme. It is required for the enzyme to function. So why do we need vitamins? Some vitamins help the body to make coenzymes. Some vitamins actually are the coenzyme. So you may have heard on commercials, I hear that all the time, CoQ10. CoQ10 is a coenzyme Q10, and it helps to make ATP and prevent DNA damage. So vitamins are important to us. Some of them help the body to make coenzymes, and so we need coenzymes in order to have reactions take place. Some vitamins actually are the coenzyme. CoQ10 is an example. It helps to make ATP and prevent DNA damage. All right. Next, left-hand side page, we're going to have data for a little activity, so just leave that blank. On the right-hand side, some important information. All right, so we have two different types of enzymes that are related to ATP. ATP synthase, synthesis, we're building things, we're creating things. So ATP synthase takes ADP and makes ATP out of it. ATP kinase was, remember our phosphorylation cascades, we had kinases in there. So those kinases breaking it down, it's changing ATP and ADP by breaking off one of those phosphate groups. Enzymes are not used up in a reaction. They can be used over and over and over again. But there are some things that can affect enzyme action. It can have an effect on their efficiency. So some factors that affect enzyme action: inhibition, the enzyme concentration, the substrate concentration, and denaturation. So inhibition, we looked at that before: competitive, non-competitive. It's inhibiting, it's not allowing the enzyme to work. So enzyme concentration can affect enzyme action. The more enzyme there are, the more work can be done. Substrate concentration, if the substrate's really dilute, it might be harder for the enzyme to get anything done because it's, waiting to bond with one of those substrates. Denaturation, that's when the enzyme undergoes conditions that causes its folding to change. It gets it out of whack. So if you get it too hot or too cold, it's changing the folding of that enzyme, changing the folding of that protein, a change in shape, change in function. pH, too acidic, too basic, can denature it. Salinity, if there's too much salt or solute in a solution, it can denature it. All right, so on the bottom half, let's do this part first. Free energy is the energy available to do work, also known as activation energy. Free energy is the energy available to do work. It's also known as activation energy. An exergonic reaction is a spontaneous chemical reaction resulting in a net release of energy. So these are a couple of important words here. Spontaneous means it can happen on its own. Net release of energy, sometimes, there needs to be a little bit of energy input, and then there's going to be a lot more energy that's released. So exergonic reaction is a spontaneous chemical reaction resulting in a net release of energy. Endergonic reaction, non-spontaneous chemical reaction. That's RXN. That's my abbreviation for reaction. Write it out if you're not sure. A non-spontaneous chemical reaction where energy from the environment is absorbed. Exergonic, exit, energy is released, the net release of energy. Endergonic, into, energy from the environment is absorbed. Activation energy is the amount of energy required to start a reaction. So in order to get something going, to get a reaction going, we do have to input some energy. So this first diagram is sort of looking at that activation energy with and without an enzyme. So the black line is showing the course of a reaction without an enzyme. It requires some energy input in order to get the reaction going. So this represents the progress of the reaction. Got to input some energy. When we have an enzyme, the amount of activation energy that we need is lower to get that reaction running. So black line represents without the enzyme, red line represents with the enzyme. So notice that we don't need to add as much free energy to get that reaction going when we have an enzyme. It's helping to get the reaction going. It's acting as a catalyst. Enzymes act as catalysts in reactions. And so the other diagram just gives us a nice visual of the two different types of reactions. So exergonic, energy is released, it's spontaneous. So here's my reactants. Energy is released. Here's the amount of energy left in the products. We have this much energy released. Endergonic, non-spontaneous. We need to input energy in order to create the products. So here's graphic representation of how much energy needs to be absorbed from the environment in order to create those products.